Here we have what is a typical prokaryotic DNA strand. So most prokaryotic DNA strands have what is called a promoter region. This promoter region is highlighted in yellow. And here you can see that there is a -35 box and a -10 box. These two are very important for the RNA polymerase to recognize and bind onto that part of the DNA. The -10 box is important because it is essentially what the RNA polymerase recognizes as where it should attach to. So if this gene were to be responsible for creating an enzyme that can metabolize sugar, if it didn't have the -10, then the RNA polymerase would not be able to bind to this DNA and create the RNA necessary for that enzyme. Therefore, that gene would never be expressed because that enzyme could never be produced. So the RNA polymerase itself is made up of two parts, the core RNA polymerase and the sigma factor subunit. The sigma factor subunit is what recognizes the promoter region specifically. And so once the sigma factor subunit attaches to the promoter region, the RNA polymerase will also bind, creating a hollow enzyme. So as a whole, it is called the hollow enzyme. And so as you can see, it binds to the promoter region and it moves down. And as it gets to the +1 box, that is where transcription begins. And so the RNA polymerase is responsible for unwinding the DNA, separating the template strand from the coding strand, and allowing the RNA polymerase to move down and transcribing the DNA into RNA. Now the top strand is called the coding strand, and this is not what is being transcribed, but the strand on the bottom, the template strand, is what is transcribed. So the coding strand is usually flowing from 5' to 3', and the template strand is from 3' to 5'. So as the RNA polymerase moves from the 3' to the 5' on the template, it would create RNA strand that is from 5' to 3', with each unit being added to the 3' end of the RNA. So when the RNA polymerase attaches nucleotides, it is in the form of ribonucleoside triphosphate. And so these have 3 phosphate groups, and as each nucleotide is added, complementary to the template strand, 2 phosphate groups are removed. And this creates a phosphodiester bond between each nucleotide that is added. So here you can see that the complementary RNA to the template strand has been created from the 5' end to the 3' end. Now, how do RNA sequences usually terminate? So one way for it to end is what is called a row-independent termination that occurs from an inverted sequence within the DNA. So within this inverted sequence, this creates an RNA strand where the nucleotides bind to each other, creating a hairpin formation like so. Then further downstream of this RNA, there is what's called a poly-A tail, where the DNA has a consecutive sequence of multiple adenines. This would create complementary uracil tail, and this A to U link is very weak, so this dissociates really quickly. So once the RNA dissociates, then that is the end of transcription.